Good day, good day, good day, good day. Welcome to our first lesson. Today I'll be covering the topic called genetics. It's mostly taught in term 2 for grade 12 life science learners. So without wasting any time, I'm going to go straight to the introduction and what to expect, sort of typical questions based on the content we'll be covering today. So the first question that comes to mind that I always get from people is why do we study genetics? What makes genetics special or unique? For example, every human being, even animals, for example, um, the traits that they have tend to be passed to their offspring. For example, most children tend to look like either their parent. Some look more like their fathers, some look more like their mothers. Some even look more like their grandparents or their great parents. So with genetics, it can allow us to understand how the traits are being passed from parents to offspring. And also, if there's a change, for example, maybe there's a disturbance in the DNA sequence, things such as mutations, why are kids being born with sort of like syndromes? So with genetics, we can be able to track, for example, Down syndrome. We can track the chromosomes and check the complications which are arising due to chromosomes. And for such, we can use genetics. So it's very important to study genetics because we can track and trace different complications or different traits which are being passed to from parents to their children. And also evolution, which is another topic that is covered also in grade 12. Most people, they always say, or as human beings, we are from apes, and then evolution occurred and stuff like that. So if we test or we track or we check the DNA sequence or the amino acids which are part of also from the DNA sequence, because amino acids come from DNA sequence before we undergo transcription, it was a DNA first. So with tracking the DNA sequence or studying genetics, we can track the differences or the similarities between primates or also human beings. That's why genetics is very important. I always tell people this is a very important topic to study. You can even end up studying disorders which can lead you to become a counselor, or you can use it, for example, police officers, they can use genetics to track or to check or to find the criminals, which we're also going to cover as we do other sections later on. So we've got what we call nucleic acids, and then we've got two of them in our grade 12 syllabus that I'm going to focus on now. We've got DNA and also RNA. So you must always know that DNA and RNA, they are made up of building blocks or monomers called nucleotides. So I've seen most questions where they're asking what are the building blocks or monomers of nucleic acids. So it's the nucleotides. So nucleotide has got three different regions. The first one, I'll start with the phosphate group. This region here, the phosphate group. So the phosphate group is common in both DNA and also RNA. And then we also have got the sugar. So you guys must always know that the sugar in RNA is ribose. The sugar in DNA is deoxyribose. That's the major difference between the sugars. The sugar in DNA, deoxyribose. The sugar in RNA, ribose. And then lastly, we've got these guys, which are four, the nitrogenous bases. So we've got four in both DNA and also RNA. In DNA, we've got adenine, cytosine, guanine, and also thymine. That's in DNA. We've got adenine, cytosine, guanine, and also thymine. Those are the four that we have in DNA. And yeah, I apologize here, it's guanine. There's an E there. And then also after that, it's an end, which should be afterwards. So we've got those four in DNA: adenine, cytosine, guanine, and also thymine. And In RNA, this is the other difference that you guys must become aware of. In RNA, there is no thymine in RNA. Instead, we've got uracil. So in RNA, we've got uracil. In DNA, we've got thymine. That's another difference that you must become aware of. And then those are the two differences that are going to come from this section so far. Is the thymine and also uracil, and then also the sugars, which are also different from the two of them. So also quick history about the discovery of DNA. So far, from what I've read so far, we had Rosalind Franklin, who discovered or had X-ray pictures of DNA, and then apparently her assistant shared the pictures with Watson and Crick, and then this eventually led to Watson and Crick publishing or doing and sort of like a research about the double helix structure and also the pairing, which is still today a very important concept that Watson and Crick published. So most of the time they always ask a question, especially in that section of A only, B only, A and B, where they're asking about people who published or discovered about DNA. So it's mostly Watson and Crick. So they got a Nobel Prize in 1962 about the discovery of DNA. So they also mentioned the very important part about complementary base pairing, which we'll cover in the next few minutes. So let's go to DNA now. First of all, what is DNA? DNA is in full it's deoxyribonucleic acid. It's deoxyribonucleic acid. So the D stands for deoxyribonucleic acid. A is acid and then N is nucleic. So the D is deoxy, N nucleic, and then A acid. So mostly it's found in 3 locations. From grade 10 life science, they told us that DNA is found in the nucleus, which we will focus mostly on when we do protein synthesis and also when we do chromosomes and karyotypes later on. So the DNA that is found in the nucleus is called the nuclear DNA. And then also you can find some DNA in the mitochondria. We're going to also find the Y chromosome, which we also will talk about when we do things like out of Africa hypothesis later on. So DNA in mitochondria is called mitochondrial DNA, and then also there's some small amount of DNA which can be found in the chloroplast, which is called the chloroplastic DNA. So as I mentioned before, we've got 4 nitrogenous bases in DNA, which are adenine and also guanine. So you need to know that adenine and guanine, they call them the purines or purines. And then cytosine and thymine, they call them the pyrimidines. So whenever you see a question where they're asking you for the name of adenine and guanine, is the purines, while cytosine and thymine is pyrimidines. So now let's go to how the structure will look like. So remember Watson and Crick, they discovered about the pairing. So we must always know adenine will always pair with thymine. They always pair together, adenine and thymine, that's in DNA. And you must always know the weak hydrogen bonds will be found between this pairing. We've got what we call weak hydrogen bonds that will hold the 2 bases together. So between adenine and thymine, we've got 2 bonds. And then in between guanine and also cytosine, that's when we get 3 bonds. That's another difference that you guys must become aware of. Between adenine and thymine, we've got 2 bonds. Guanine and cytosine, we've got 3 bonds. So the structure is the double helix. So always remember that the DNA will always coil around each other. It is double stranded. So it's got 2 strands. So DNA is double stranded because it has got 2 strands. So that's how the structure will look like. Remember, the nucleotide will be phosphate group. This will be the phosphate group. Then we've got the sugar, which will be the deoxyribo sugar more, and then we're gonna have the bases. And then that's it about the DNA. So let's quickly go to RNA. Remember now, DNA is double helix or double stranded. Now RNA is single. Remember, DNA is coiled, RNA is not coiled. And then also RNA is shorter. And then remember the sugar also, and the difference. In DNA we've got deoxy, ribo, and then in RNA we've got the ribose sugar. And the difference in the nitrogenous bases, DNA we've got thymine, and then in RNA we've got the uracil. So for example, we know in DNA, adenine always pairs with thymine, but here in RNA, adenine will pair with uracil. So always become aware of that because of the change. There's no thymine in RNA, it's uracil. This is very crucial when we are forming RNA for protein synthesis, which we'll learn about later on. So there are three types of RNA, which I will also going to talk about when you talk about protein synthesis. The first one is the messenger RNA or the mRNA. So it's mostly found in the nucleus. And then it mostly leaves the nucleus to go to the cytoplasm so that it can meet with this guy or girl called transfer RNA. Transfer RNA is also going to be found in the cytoplasm, and then it mostly comes with anticodons, which will complement with the codons of messenger RNA. Then they're going to form amino acids. Then the last one is the ribosomal RNA, which is also found in the cytoplasm. So please guys, always become aware of these differences, because they always ask questions most of the time. Tabulate the difference between RNA and DNA, for example, for 6 marks or 7 marks. So such questions must always be there. Remember, this will be under difference, under difference, under difference, under difference. They can say tabulate 3 differences or 4. So always become aware of such questions. So that's it. I wanted to add questions today, but I feel like no, let's just do the sections as they are, and then once I've covered protein synthesis, then I will do different past papers, maybe 3 or so, and then we're going to be tackling these questions together and see how to approach them and then how to answer them. And then yeah, thank you very much for this section, and then yeah, next time.